Welcome everyone, and thank you for virtually joining us for the latest version of the Variant Effects seminar series. My name is Diego, and I'm a member of the VEST organizing committee, along with Yanil Budu and Adrini D'Souza. These seminars are made possible with support and a lot of help from Laura Muffley and Alex Hopkins, among several others, and our sponsor, the Atlas of Variant Effects Alliance. So a huge thank you to them. As a reminder, we're always on the lookout for potential speakers for future seminars. If there's anyone you'd like to see present, or if you're interested in sharing some of your own work, then feel free to submit a speaker nomination form. As usual, before these presentations, I have some housekeeping items to mention. Each talk is about 20 to 25 minutes, so there should be time for a few questions at the end. Please post those questions at any time to the Zoom Q&A, and we'll read them to the speaker at the end of their seminar. If you have additional questions after the end of the seminar and you're an AVE member, feel free to visit our seminar series Slack channel for more discussion. And finally, we're posting updates on our Twitter account, so be sure to follow us there at Variant Effects. You can catch the seminar today and previous presentations that have been recorded on our affiliate CMAP YouTube channel. I'm going to post all these links to the chat once we get started. All the details of this seminar series can be found on our website, including a regularly updated schedule of upcoming speakers. And for the last item, this is rarely an issue, but just as a reminder, please be considerate when posting comments or questions. We expect everyone to be respectful and use welcoming and inclusive language. So without further ado, we have two excellent speakers scheduled for today's presentation. Starting us off, Megan Buckley worked with Greg Finley at the Francis Crick Institute on using high-throughput functional genomics assays to understand the effect of pathogenic alleles. She's now pursuing her doctorate at Cambridge with a focus on chromatin and epigenetics. The title of Megan's talk today is Resolving the Functional Spectrum of Pathogenic Alleles Across VHL. Megan, go ahead and share your screen and start whenever you are ready.
Yeah, thank you for the introduction, and thank you to all the organizers for putting on the seminar series and for inviting me to speak today. So as Diego said, I'm going to be presenting on my work that I carried out at the Francis Crick Institute in London, where I looked at the functional effects of single nucleotide variants in VHL. So VHL, or von Hippel-Lindau, is a tumor suppressor gene with 3 exons, and it encodes an E3 ubiquitin ligase that's involved with the hypoxia response. And it does that through HIF1 alpha, which is its canonical target. Somatic mutations in VHL are common in renal cancers, particularly in clear cell renal cell carcinoma. And in fact, RCC is estimated to make up to 70% of all renal malignancies. Germline pathogenic variants in VHL cause VHL disease, which is a cancer predisposition syndrome, where these patients are susceptible to multiple different tumor types, including RCC, hemangioblastomas, and pancreatic and renal cysts, to name a few common ones. The disease is classified into type 1 and type 2A, B, and C. Type 1 disease patients have a high risk of developing ccRCC and also develop hemangioblastomas and cysts, whereas type 2B additionally is characterized by the risk of pheochromocytomas. So type 2A patients have a risk of hemangioblastomas and pancreatic cysts, and type 2B also has the additional risk of ccRCC, whereas type 2C has a pheochromocytoma-only phenotype. And RCC occurs in around 70% of VHL disease patients by the age of 60 and is the leading cause of death. So given the diversity in phenotypes and the link of different variants to different subtypes of disease, it's still unknown why these different variants cause different tumors. The molecular basis of the disease isn't very well understood. So what is known is that renal cancers seem to lead to loss of HIF regulation, which, as I said, is VHL's canonical target. HIF can then migrate into the nucleus and activate downstream pathways involved in oncogenesis, like angiogenesis and glucose transport. Whereas for pheochromocytomas, it's thought to act on HIF-independent mechanisms. In the clinical database ClinVar, there are over 800 VHL variants of uncertain significance, or VUS. And there are still many more variants that haven't yet been seen clinically, that haven't been reported in ClinVar. And even for the known pathogenic variants, it's still difficult to interpret a tumor risk profile, as there's discrepancies in reporting, and also often the disease type isn't specified. So in order to further understand how VHL disease mechanisms work, I used saturation genome editing to functionally assay all single nucleotide variants in VHL. So saturation genome editing, or SGE, is a high throughput method that allows you to assay the functional effects of single nucleotide variants within a genomic region of interest. This experiment can be done in multiplex, like using CRISPR to integrate variants such that you get one variant per cell. And if the gene is essential in the cell line, then loss of function will lead to cell death. And you can then calculate what we call a function score, which compares the abundance of variants across different time points. So for VHL, I designed 7 SGE regions, which tiled across the whole coding region of the gene, and included an intronic region as well. I also performed these experiments in hap1 cells, which have a haploid genome, which meant that for every variant I was testing, I was only seeing the effects of the variant allele, and the effects weren't being masked by the wild type allele. Initially, when I did the experiments, only subtle effects were seen compared to what we'd seen previously in our work in BRCA1. So I made some optimizations to the protocol so that we could specifically assay variants in VHL. Firstly, I allowed 20 days of experimental time course to see the effects fully. And I also improved library design and preparation and modified the transfection protocol, which improved transfection efficiency, library quality, and editing rates. And I obtained function scores for more than 2,200 single nucleotide variants across the entire gene. In particular, the addition of a compound called DAB really improved the range of function scores that we saw. So DAB is actually a precursor to the chemotherapy drug Taxol, but it was identified in this paper I referenced down here as a compound that could enrich for haploid cells. When I added it into our cell culture medium, I saw the same effect. And we also saw a big improvement in the separation of canonical splice variants and nonsense variants in our data. To define loss of function variants, we calculated a null distribution defined as synonymous variants that didn't impact on RNA, or for exon 1 5', this region here, which didn't have RNA scores. It was simply all of the synonymous variants that were tested. As the intronic region didn't have any synonymous variants, we fitted a normal distribution to all of the variants tested in that SGE region. And with a false discovery rate of 0.01, 16% of single nucleotide variants scored loss of function by this definition. Plotting the function scores against the chromosome position allowed us to generate what we call these variant effect maps. And each variant here is represented by a dot and coloured by the consequence that that variant causes. Interestingly, there was no functional consequence of variants upstream of methionine 54, which acts as an alternative translation start site. But in the rest of the gene, all of the nonsense variants were significantly depleted, and also splice variants were too. So this kind of showed us that our assay was working well, since you'd expect splice and nonsense variants to cause loss of function. Most missense variants in green scored neutrally. 23% were loss of function in the assay, and these seem to align to secondary structural features like the beta sheets and alpha helices that I've mapped above the plots here. We looked at gene essentiality on the cancer dependency map, which is an online tool, and we used a data set that did a genome-wide CRISPR screen for more than 1,000 cell lines. So looking at the Cronus scores here, you can see that VHL loss in cell lines with no VHL mutations reduced cell growth, whereas cell lines with VHL mutations, which were generally kidney derived, were more tolerant to loss of VHL. And since HAP1 cells are wild type of VHL, this meant that we could use a growth phenotype to assay the functional effects of variants. So to try to understand why VHL loss led to reduced cell growth in the cell line, I performed a series of knockout experiments to identify the downstream pathways that were being represented here by VHL loss. So VHL is upstream of a few quite major signaling pathways, including the RAF-UC pathway and NF-kappa B pathway. So as you can imagine, there's potential to have many downstream effects. So first here, we've just got some cell images with a VHL only knockout. And I did a double knockout with VHL plus the housekeeping gene HPRT1. And as you can see, they look very similar. And I did VHL plus another protein double knockout for many different proteins. And the only one that had growth phenotype was VHL HIF1A double knockout, which seemed to kind of rescue the growth phenotype compared to the controls. So I generated a HIF1A knockout cell line and repeated SGE for around 800 single nucleotide variants across exons 2 and 3 of VHL. And as you can see in the HIF1A knockout cell line, all variant effects were eliminated by the loss of HIF1A. When I compared the function scores across the HIF1A knockout cell line and the wild type cell line, there's no relationship between function scores. And you can see the difference in axis scales here as well. So this indicated that the SGE function scores specifically reflected the HIF-dependent effects that VHL SNVs cause. Thinking about the mechanisms by which the SNVs cause loss of function, we decided to look at the effect of the variants on RNA levels. So we derived RNA scores by extracting the RNA at day 20 and day 6 and comparing the abundance of the RNAs, the variants compared to the abundance in genomic DNA. And the RNA scores reflect the SNVs effects on full-length VHL mRNA levels. So you can see that many SNVs with that caused low RNA levels were at splice junctions, which means that they're likely to disrupt splicing, as you'd expect. But we also saw some other interesting variants, like this synonymous variant here at coding position 222. which is actually been reported to cause a severe deficiency in VHL at birth without showing a phenotype in the parents or carriers. This had a function score of minus 0.4, which is slightly reduced, but wasn't significant with the FDR of 0.01. But this just highlights that this aspect of our data analysis could be useful for identifying rare variants like this one. When we compared the RNA scores to our function scores, we saw that most loss of function variants actually had didn't deplete in RNA and had quite neutral RNA scores, and a substantial reduction in RNA levels was required before the reduction in RNA caused loss of function. Also, if you look at the nonsense variants in red, they're minimally depleted. So we concluded that there was a weak nonsense mediated decay effect. So that this data does suggest that a minimum RNA level is required for normal cell growth. And the functional effects seen here are not dependent on RNA. When we mapped the function scores to the 3D structure of the protein, we saw that they, again, mainly map to alpha helices and beta sheets. When VHL functions as an ubiquitin ligase, it does so in complex with the longins B and C and culin 2. And looking at where the lowest scoring variants localized to, we saw that many of the lowest scoring variants occurred at the interface between VHL and login C, and also at the HIF1A binding site. So this confirmed to us that the functional effects seen in our SGE assay were likely to be exerted at the protein level because it would impact VHL's ability to bind to its targets and interactors. Looking at how well function scores predict disease pathogenicity, I've colored the data here by the classification the variant has been given in ClinVar. And you can see that pathogenic variants separate quite nicely from those classified as benign or likely benign. And they did so with 95% sensitivity at 98% specificity. In comparison to variants present in population databases, which scored neutrally. So that meant that the majority of rare variants that are seen multiple times in the population are unlikely to cause disease. So which variants are seen in human cancers? Well, given that many of the rare variants usually classified as VUS in the clinic, and they could arise spontaneously in tumorigenesis, we used the C-Bioportal cancer database to define variants associated with clear cell renal cell carcinoma. And when we looked at this, we saw that our function scores separated all of the CC-RCC variants in C-Bioportal from those classified as benign or likely benign in ClinVar or present in population database. And in fact, 22% of variants in other tumors were loss of function in our SGE data, which is slightly higher than the fraction of all variants at 16%, but these we thought could be passenger mutations. So it seems that loss of function variants are driving mutations in RCC. So with a high accuracy that function scores predict these pathogenic variants, we hope that the data could be useful for making clinical decisions. With this in mind, and the fact that only 3 messenger variants are classified as benign in ClinVar, and over 75% of variants present in the database are classified as variants of uncertain significance or have conflicting mutations, we needed another way to define a good standard set of cancer missense variants. So the set that we defined includes variants that are seen in at least 2 independent clear cell renal cell carcinoma cases. Or if they are only present in one case, they would be classified as pathogenic or likely pathogenic in ClinVar. And our function scores separated this set of variants with perfect accuracy from those that were benign or likely not benign in ClinVar and present in at least one population database. And since ClinVar lacks benign and likely benign missense variants in VHL, the function scores also separated the 73 missense variants in the gold standard set from the 99 missense variants that were not deemed pathogenic or likely pathogenic in ClinVar and were present in at least 2 population sequencing controls. So these analyses indicate that SNVs scoring below a threshold of minus 0.478 are likely to promote CCRCC, whereas those scoring above minus 0.288 do not cause CCRCC. We also compared our SGE data to computational predictions, and when the 5 computational models that we looked at all predicted more deleterious missense variants than what is suggested in our SGE data. Some of these could be type 2 variants, so those with the risk of pheochromocytoma that aren't mediated through HIF-dependent effects. But this is quite difficult to prove since these effects are subtle. But this analysis still showed that the computational models lack specificity for disease phenotype. However, in general, there is broadly an agreement between models in support of the variants which scored loss of function by SGE. One challenge still remains, and that is being able to distinguish type 1 and type 2 variants. So we're currently exploring this further, but we think our data reasonably well supports being able to classify a variant as type 1, type 2A, type 2B, or type 2C, as the scores seem to correlate quite nicely along the spectrum, and the degree of HIF regulation is a major determinant of the clinical manifestations of the disease, and our evidence supports this. So as you can see, the type 1 variants in general do seem to score lower than the type 2 variants, which seem to score intermediately. So yeah, we think that our data explains the diversity of phenotypes seen in VHL disease. So to summarize, I optimized an SGE assay to measure the effects of nearly all possible single nucleotide variants across VHL, which meant we could accurately predict whether variants were associated with disease, including defining a gold standard set of CCRCC variants. And the variant effects that we saw reflected the HIF-dependent mechanisms in VHL disease, and the variants mainly exerted effects at the protein level. But for patients harboring germline VUS in VHL, our data may provide valuable evidence in support of variant classification or reclassification in some cases, alongside ongoing trials for the HIF2A inhibitor belzutifan, which is in trials to treat VHL disease. But beyond this, we anticipate that the function scores may help clinicians predict tumor risk given a patient's specific variant. So the most important slide, I just want to thank Greg and the rest of the Findlay lab, everyone who contributed to the work. I now have a preprint out about this, so please check that out if you want to find out more. and thank you everyone for your attention.
Good. Wonderful. Thank you so much, Megan, for this great presentation. We can start with questions. I see one from the audience. So, did you identify any VHL gain of function variants?
So in this particular assay, we were just looking at loss of function variants, but for sure you can make changes in the assay if you want to identify gain of function variants. Yeah, but in this work, we didn't look at the gain of function though.
Okay. So we have another question. So from Adrini, so the VHL effects seem to be dependent on if A1, but not EPAS. So if2A, sorry.
Mm-hmm.
In your cell knockout experiment, how do you view the contribution of each HIFs to disease, which is more important for RCC?
Yeah, that's a really good question. Thank you. So yeah, I don't know if you noticed, but I did also look at VHL with HIF2A double knockout, and we didn't seem to see any effects. And I think HIF2A is also a target of VHL, but HIF1A is its canonical target. And yeah, from what we saw from our data, it seemed that the effects seen were specifically HIF1A dependent rather than HIF2A. But yeah, it would be interesting to see if HIF2A was also maybe involved more in pheochromocytomas or other renal related diseases.
Well, I'll ask one more question. So does it matter which cell line you use to study VHL?
No, that's a good question. So as I mentioned, we chose HAP1 cells because they have a haploid genome. And we know that there's no VHL mutation in HAP1s. And it also was easy to then only see the effect of the variant that we were looking at. But yes, there is not for VHL, but for other genes, there is work in the lab looking at different cell lines. So you can for sure adapt an SG protocol to look at the effects in different cell lines. But I suspect that you would probably see similar effects. But yeah, for VHL specifically, often in RCC, VHL is completely lost. So that's why HAP1s were good specifically for VHL.
So one last question from the audience. So were these experiments done at atmospheric oxygen levels? And do you think you would see an increase in growth for pathogenic mutants at physiological oxygen levels?
Yeah, so the cells were just cultured in kind of typical cell culture conditions. But yeah, I think one really interesting way to kind of further explore the HIF-mediated effects might be to kind of induce normoxia, and look at in vivo oxygen levels as well.
Okay, thank you so much. Thank you very much.
Thank you. Thanks.